Welcome to the Deep Dive. I'm your host, and today, well, we're taking a stack of really critical scientific sources. We're going deep, especially into Chapter 230. Our goal, to pull out the most vital insights, the kind of stuff that gives you a truly professional-level grasp of a fascinating and often hidden world. We're talking about parasitic infections.
That's right. It's a whole hidden ecosystem, really.
Exactly. And we're not just scratching the surface today. We want to understand the intricate lives of these organisms, from the tiny single-celled invaders right up to the macroscopic parasitic worms.
And the mission, as you said, is to really understand their complexity. What's captivating, I think, is the sheer ingenuity of these pathogens and the profound impact they have, often in ways we don't immediately see, on global health.
Right.
Uncovering their unique biology, how they live, how they thrive, that's absolutely key to fighting them. It's the foundation.
That's the core of it. So our mission today, go incredibly deep into this material with a special focus, as we said on Chapter 230. We want to arm you, the listener, with a professional-level understanding. Protozoal infections, helminthic infections. Let's unpack this.
Demystifying Trichomoniasis, a protozoal primer.
Okay, so let's start this journey, this exploration, with an organism maybe you've heard of, but perhaps don't know the full story behind. Trichomonas vaginalis. It's actually one of the most prevalent protozoal parasites right here in the United States. But it often, flies under the radar a bit. What exactly is this little single-celled invader?
Yeah, let's define it precisely. Trichomonas vaginalis, it's a very distinct organism, pear-shaped, actively motile, meaning it moves around purposefully, and it's a single-celled protozoan. When we say single-celled, don't let that fool you into thinking it's simple in function. It's incredibly efficient. It's size-wise, we're talking roughly 10 by 7 micrometers.
Really tiny, about the size of a red blood cell.
Exactly, right on that edge of microscopic visibility. And it replicates very, very efficiently using binary fission, basically just splitting itself in two.
Simple division, but effective.
Incredibly effective, and that's key to how it establishes large populations within a host. Now, its primary habitat, where it really likes to live, is the lower genital tract in females. And in males, it tends to set up shop in the urethra and sometimes the prostate. This specific preference, this tropism for the genitourinary system, well, that fundamentally dictates how it spreads and the diseases it causes.
That specialization is interesting.
It is, and it's worth just briefly mentioning, while T. vaginalis is our main focus because it's the major genitourinary player, there are other Trichomonas species. For instance, you might find Trichomonas tenax in the mouth, often linked to poor oral hygiene or periodontitis. And less commonly, you might find species like Pentatrichomonas hominis in the gut. But the one that really grabs our attention for public health is T. vaginalis, precisely because of its specific impact on reproductive health. So the takeaway, this isn't just any random microbe, it's a highly adapted specialist.
Right, a specialist of the human genitourinary tract.
And that has huge implications for how it spreads and how we control it.
Okay, that specificity is key. But it's still just a single cell. Yet somehow it becomes incredibly widespread, affecting millions. How does that happen? How does it move from person to person so effectively? Let's map out its life cycle and epidemiology.
The prevalence numbers really paint the picture, don't they? In the U.S. alone, estimates are around 3 million infections per year in women.
3 million. That's not trivial.
Not at all. It immediately tells you this is a major public health concern, not some niche infection. And when you look at how it transmits, well, it's overwhelmingly direct person-to-person venereal transmission.
So almost exclusively sexually transmitted.
Almost exclusively, yes. The sources are very clear on this. While, theoretically, the organism can survive for a few hours in a moist environment, maybe a damp towel, perhaps a hot tub, though that's rare, direct intimate contact is really the dominant mode by far.
Which means control strategies have to focus there.
Absolutely. They must be centered on sexual health strategies. And there's another critical piece to the epidemiology. It's strong link to other risk factors. We see its prevalence is highest among people with multiple sexual partners, and importantly, also among those who already have other sexually transmitted diseases.
So it clusters with other STIs.
Exactly. It's not just a random correlation. It points to shared vulnerabilities, shared risk behaviors. This means you really have to look at trichomoniasis within the bigger picture of overall sexual health and STI prevention.
That makes sense. Addressing one might help with others.
Precisely. Tackling trichomoniasis can often shed light on or even help mitigate risks for other infections as well. It's all interconnected.
That interconnectivity is crucial. Yeah. Okay, let's pivot now to the clinical side, because this is where things get, quite nuanced. You mentioned the presentation can be really different between men and women. That seems like it would have massive implications for diagnosis and also for stopping the spread. What should professionals be looking for?
You've hit on a really central challenge, both clinically and for public health, this asymmetry in how it presents. And in males, honestly, it's often insidious. It can be quite deceptive. A significant chunk of men infected with T. vaginalis are completely asymptomatic, no symptoms at all.
So they can carry it and transmit it without knowing.
Exactly. They carry it, they can pass it on, but they feel perfectly fine. This silent carrier state is a huge hurdle for control efforts because these individuals are unknowingly keeping the transmission cycle going. Now, while many are asymptomatic, some men do develop symptoms. Typically, urethritis inflammation of the urethra. That might mean painful urination or maybe a discharge. Less commonly, it can lead to more serious issues like epididymitis, inflammation of the tube behind the testicle, or prostatitis, inflammation of the prostate.
Okay.
So the key thing for professionals working with males, absence of symptoms absolutely does not rule out infection. And even when symptoms are present, they can easily mimic other conditions.
Got it. A high index of suspicion is needed. What about in women?
It's a different story in women. Infection is usually, though not always, symptomatic. The incubation period, the time from getting infected to showing symptoms, typically ranges from about 5 to 28 days. The classic symptoms we look for in women include a malodorous vaginal discharge, often described as yellow-green, sometimes frothy. Also, vulvar erythema, which is redness and inflammation of the external genitals, and often intense itching.
Okay, sounds uncomfortable.
Very. Dysuria, or painful urination, and increased urinary frequency are also quite common. Maybe 30 to 50% of patients experience these. And dyspareunia, painful intercourse, can also be a significant issue. But, and this is a really critical diagnostic point, these symptoms, even the classic ones, do not reliably distinguish trichomoniasis from other common causes of infectious vaginitis.
Like bacterial vaginosis or yeast infection.
Exactly. BV, candidiasis, the symptoms can overlap significantly. So relying on clinical presentation alone is just not enough for a definitive diagnosis.
No.
You absolutely need precise laboratory confirmation. Otherwise, you risk misdiagnosis and giving treatment that won't work.
So that leads directly to the next question. If the symptoms are often ambiguous, especially with asymptomatic men and overlapping symptoms in women, how do clinicians nail down the diagnosis? What are the gold standards? And what are the real game changers in diagnostics lately?
That's the crucial question, isn't it? How do we confirm it? Historically, and actually still used quite a bit because it's fast and cheap, especially in places with limited resources.
Yeah.
The go-to method has been microscopic examination of wet mounts. You take a sample of vaginal or maybe prostatic secretions, put it on a slide with saline, and look for those characteristic motile trichomonads swimming around.
Sounds straightforward. Immediate results.
Immediate results, yes. But here's the major drawback. Its sensitivity is notoriously low. We're talking maybe 50% to 60% sensitivity in routine evaluations of vaginal secretion.
Only 50 to 60%. That's quite low.
It's a significant diagnostic gap. It means you could be missing nearly half of the infected individuals. That leads to ongoing transmission, potential complications. It's a major barrier to effective control, not just an inconvenience.
Wow. Okay, so what's better?
Well, to get around that low sensitivity, more advanced methods came along. Direct immunofluorescent antibody staining, or DFA, is one that offers better sensitivity, maybe in the 70% to 90% range. DFA uses fluorescently tagged antibodies that stick specifically to the parasite, making them easier to spot under a special microscope.
Better, but still not perfect.
Better, but the real state-of-the-art, the technology that has truly revolutionized diagnosis in this area, is nucleic acid amplification tests. NATEs. These are FDA approved, and they're characterized by exceptionally high sensitivity and specificity.
How do they work?
They work by detecting the parasite's unique genetic material, its DNA or RNA. This means they can find an infection even if there are only a very small number of organisms present. It's a total paradigm shift. It allows us to detect infection in those asymptomatic carriers, and it's much better for diagnosing men where it's always been harder.
And they work on different sample types.
Yes, that's another advantage. NEAT-ATs are great for urine samples, which are easy to collect, and also for endocervical and vaginal swabs from women.
What about diagnosing men specifically? Still tougher.
It is still generally more challenging in men, yes, but T. vaginalis can be recovered from the urethra in men. And you can sometimes improve the detection rate by collecting a sample after prostatic massage. That helps flesh out any parasites hiding in the prostate or urethra. But even with that, The difficulty in male diagnosis really highlights why highly sensitive tests like gnats are so incredibly important.
So gnats are really the way forward.
Absolutely. Their widespread adoption is a massive leap forward. It allows for much more accurate identification, which means more targeted treatment, better control, breaking those transmission chains. It's all connected. This isn't just lab work. It impacts public health directly.
Okay, so we have the diagnosis locked down with something like a gnat. What's the best way to treat it? And what are those broader considerations beyond just the individual patient? How do we make sure we're not just clearing the infection temporarily, but actually preventing recurrence and impacting public health?
Treatment itself, once you have that accurate diagnosis, is usually quite effective. But as you say, it requires a strategic mindset, not just focusing on the one person. The main drugs are metronidazole and tinidazole. Both are in the nitramidazole class.
And how are they given?
Metronidazole can be given in two main ways. Either a single large 2-gram oral dose, which is very convenient for compliance, or as 500 milligram doses taken twice a day for 7 days. Tinidazole is usually given as a single 2-gram oral dose. Both of these regimens are generally highly effective at getting rid of the infection in the patient sitting in front of you.
But that's not the whole story, is it?
Not at all. This is where a really critical public health principle comes into play. It is absolutely essential, imperative, really, that all sexual partners of the infected person must be treated at the same time, concurrently.
Not optional.
Absolutely not optional. It's a non-negotiable part of successful treatment and, crucially, preventing reinfection. We talked about those asymptomatic male carriers, right? They are major drivers of continued transmission. If you treat one person but their partner remains infected and untreated, they can simply reinfect the person you just treated. You end up in this frustrating cycle of seemingly recurrent infections.
So partner treatment is key.
It's a cornerstone for managing all STIs, really. It hammers home that these aren't just individual health problems, they're interconnected, communal health issues.
Are there other tricky situations or special considerations?
Yes, definitely some nuances. For example, consider a male patient who has persistent urethritis symptoms even after getting the standard treatment for non-gonococcal urethritis. In that situation, clinicians should strongly consider treating with metronidazole for possible trichomoniasis. T. vaginalis can be an overlooked cause of those persistent symptoms. It needs to be in the differential diagnosis.
Okay, good point. What about pregnancy?
Pregnancy presents a significant clinical challenge. Metronidazole is still the preferred drug, but obviously there are concerns about medication use during pregnancy. Unfortunately, good alternatives are not readily available or might have their own issues. So treatment decisions during pregnancy become more complex, requiring a careful discussion between the clinician and the patient about risks and benefits.
And what about treatment failures? Is it always reinfection?
Reinfection is often the culprit when treatment seems to fail, absolutely. But we are also seeing, with increasing frequency, strains of T. vaginalis that show high-level resistance to metronidazole.
Oh, drug resistance.
Yeah.
That's a problem.
It is a growing problem, and it presents a real challenge. However, we do have strategies to manage these resistant cases. Options include using higher oral doses of metronidazole, sometimes using intravenous doses to get better drug levels in the body, or even using oral and vaginal doses together for a stronger local effect. Tinidazole can also be effective against some metronidazole-resistant strains, so it provides another option. It shows that even with resistance, our understanding allows us to adapt clinically.
It's really striking, isn't it? How the individual treatment plan, dealing with resistance, pregnancy, it all directly feeds into these larger public health strategies. You really can't separate the two, especially with asymptomatic carriers driving spread. It's a perfect example of microbiology shaping population health. Knowing these details is what makes an elite professional.
The World of Helminth Infections, Chapter 230 Deep Dive.
Okay, that deep dive into Trichomonas really highlights how one tiny single-cell organism can pose such huge public health challenges. But now, let's broaden our view dramatically. Let's shift to a fundamentally different, and you could argue even more complex, group of parasites. The helminths. These aren't just bigger. They operate under completely different biological rules. What exactly are helminths, and what defines this incredibly diverse group that has such a massive impact on global health?
Yeah, placing this in context is important. Helminths. They're some of the most ancient parasites around. They've co-evolved with hosts for millennia, literally shaping human history and health. So let's start with the name itself. Helminth comes straight from the Greek word helmin, which simply means parasitic worm.
Parasitic worm. Okay.
And they are incredibly prevalent organisms found all over the world. Depending on the species, they can be free-living, think of worms in the soil or water, or they can be obligate parasites, needing a plant or animal host to survive. A key feature of parasitic helminths is this long history of coevolution with specific hosts, often mammals. This has led to a high degree of host specificity.
Meaning most stick to their particular animal host.
Exactly. Most helminthic infections are naturally limited to non-human hosts. It's relatively rare, often accidental, for humans to get infected by these zoonotic helminths, the ones primarily adapted to animals.
And when that accidental infection happens?
It often results in a different kind of disease because the parasite might not be able to complete its life cycle in the human host. It can cause significant inflammation as it tries to migrate or when it dies off.
Okay, so focusing on the human ones, how are they classified?
When we look at the helminths that do regularly infect humans, they fall into two main phyla. First, the nemathelminths, these are the nematodes or roundworms. Second, the platyhelminths or flatworms. And this group is further divided into the Cestodes, which are tapeworms, and the Trematodes, commonly known as flukes.
Roundworms, tapeworms, and flukes.
Those are the main categories. And these classifications aren't arbitrary. They're based on fundamental differences in their anatomy, how they reproduce, their complex life cycles, all of which dictate the diseases they cause and how we approach controlling them. Understanding this basic taxonomy is really the first step for any professional in this field.
That lays out the groups clearly. But it brings up a point about terminology. Is every encounter with a parasite an infection? Or is there a more precise way to talk about it, especially with helminths? This feels important for clinical accuracy.
The terminology is really important here. It reflects a fundamental biological difference and impacts the clinical approach. When we talk about helminthic parasites that actually live inside the human body, in the gut, the blood, the tissues, these cause what we define as true infections.
Okay, internal parasites.
Internal parasites, exactly. Think hookworms attached to the intestinal wall or schistosomes living in the blood vessels. They've breached the external barriers and set up shop inside. But in contrast, there are other parasitic organisms, other genera, that reside only on the mucocutaneous surfaces of humans.
Like on the skin.
On the skin or mucous membranes. Good examples are the parasites causing myiasis, that's fly larvae infesting skin or body cavities, or scabies, which is caused by mites burrowing just into the top layers of the skin. These aren't considered infections in that internal systemic sense. The precise term for these is infestations.
Ah, infection versus infestation. Internal versus external.
Exactly. That distinction is critical. It highlights whether the parasite has truly established an internal presence within tissues or organs, or if it's largely remaining external or superficial. And that difference has big implications for diagnosis. Do you need blood tests or biopsies or just a skin scraping? And for treatment, systemic drugs versus topical creams. And even for public health measures, an elite professional gets this distinction right away.
That's a really crucial clarification. Okay, we just talked about Trichomonas, a single-celled protozoan. Now let's really dive into what makes helminths so fundamentally different. Because these differences, as you've hinted, are absolutely vital for understanding everything from disease progression to immune response to control. This is where we move from basic facts to deep professional understanding.
You're absolutely right. And what's truly fascinating is just how profoundly different their biological strategies are. These aren't minor details. They dictate everything. Their life cycles, how they replicate, how our immune system even sees them. Mastering these distinctions is really a hallmark of an elite professional in parasitology or infectious disease. So let's break down the key differences. First, structural complexity.
Okay.
Protozoa, like Trichomonas, are unicellular. One cell makes the whole organism. Helminths, on the other hand, are multicellular worms. And that multicellularity represents a massive evolutionary leap. It means they possess differentiated organ systems.
Like what kind of systems?
We're talking about sophisticated structures. A nervous system allowing for complex behaviors, a muscular system for movement and feeding, a developed digestive system, even if it's adapted for parasitism. This level of organization allows for much more intricate interactions with the host, more complex life stages. It's why we often call them macroparasites. You can often see them without a microscope, at least the adults.
A huge jump from a single cell. What's the second big difference?
The second one, and this is perhaps one of the most critical differences from a practical public health standpoint, concerns their complex life cycles and, crucially, their replication strategy within the host. Helminthic parasites typically have very intricate life cycles, and a key feature is that they require sequential stages of development outside the human host.
Outside the human body.
Yes, this is fundamental. They generally do not complete their replication cycle inside the human host. Instead, they develop to a certain point within us, and then as an obligatory part of their life cycle, they must mature further outside. During these extra-human stages, they might exist as free-living forms in the environment's soil, water, or they might need to pass through another host species, an intermediate host, like a snail or an insect. Only after completing these external stages do they mature into a new form capable of infecting humans again, completing the cycle.
Okay, so they don't just multiply indefinitely inside us like bacteria or protozoa might.
Precisely. And that leads directly to this paramount concept, the worm burden. This is critical. With only two important exceptions, Strongyloids stercoralis and Capillaria philippinensis, which are unique because they can internally reinfect humans.
Autoinfection, right?
Autoinfection, exactly. For all other helminths, any increase in the number of adult worms inside a person, the worm burden requires multiple and usually ongoing exposures from the outside. Exogenous exposures.
So you have to keep getting infected from the environment to get more worms.
Yes. For instance, your Ascaris worm burden only goes up if you keep ingesting more infectious eggs from contaminated soil. Your Schistosoma burden only increases with repeated skin exposure to contaminated water. This is a stark contrast to protozoan parasites.
That's so.
With protozoa, think malaria. A single mosquito bite transmitting a few parasites can rapidly lead to overwhelming infections, millions or billions of parasites, because they replicate massively within the human host. This fundamental difference, the need for repeated external exposure for most helminths to increase the worm burden, is absolutely germane, as the sources say. It's crucial for how we think about treating individuals. One treatment might clear the current burden, but re-exposure is the risk, and especially for global control efforts.
Odds of effect control.
Global programs often focus heavily on preventing re-exposure sanitation, clean water, vector control, or reducing environmental contamination, because simply treating people won't stop the problem if they just get infected again immediately. The parasite load in the community depends on continuous transmission from the environment, not just multiplication within individuals. This insight drives entirely different public health strategies.
That's a massive operational difference.
Yeah.
Okay, structural complexity, replication strategy. What's the third key distinction?
The third major difference lies in the host immune response, specifically the tendency of helminths to trigger eosinophilia.
Eosinophils.
Mm-hmm.
Those are a type of white blood cell.
Exactly. Helminthic infections have this distinct characteristic. They strongly stimulate immune responses that lead to an increase in eosinophils. You often see higher numbers of eosinophils in the infected tissues, and frequently you can measure this increase in the peripheral blood. It's a hallmark sign. And interestingly, the magnitude of this eosinophilia often correlates with how much tissue invasion is happening, either by migrating larvae or by adult worms burrowing into tissues.
So more invasion, more eosinophils.
Generally, yes. For instance, eosinophilia is often most pronounced during the early phases of several infections. Think acute schistosomiasis categorising syndrome, or during the larval migration phases of Trichomoniasis, hookworm, or Ascaris infections. That's when the larvae are actively moving through tissues, triggering a strong immune reaction.
But what about chronic infections?
Good question. In established, long-term infections, you might still find eosinophils locally around the worms in the tissues, but the blood eosinophilia might become less dramatic. It could be intermittent, mild, or sometimes even absent as the immune system kind of adapts or the worms become better contained. Also, eosinophilia is usually absent if the parasites are really well walled off, like inside an echinococcal cyst, or if they stay strictly within the gut lumen, like adult Ascaris or tapeworms, without invading the tissue itself.
So it's really a marker of tissue interaction.
Precisely. It's a response to tissue migration or invasion, and this contrasts sharply with most protozoan infections. Protozoa typically do not elicit eosinophilia. There are only about 3 rare exceptions among protozoa: the intestinal parasites, Cestodes and Dientomoeba fragilis, and the tissue-borne Sarcocystus. But for the vast majority, no eosinophilia.
So that immune signature is a really useful clue for clinicians.
Absolutely. It's not just a biological curiosity. It's a crucial diagnostic pointer that helps clinicians differentiate between potential protozoal and helminthic causes, guiding the next steps in diagnosis and treatment.
Okay, synthesizing all that.
Yeah.
It's crystal clear. These aren't just minor tweaks in biology. The differences in structure, replication, and immune response between protozoa and helminths dictate absolutely everything. how disease progresses, how we diagnose it, and critically, how we even start to control it globally. It's about understanding the enemy's entire strategy.
Right.
Let's narrow our focus now onto that first major group of helminths, the nematodes, the roundworms. You said they're incredibly diverse, found everywhere. What makes them so varied? Which ones really matter for human health, and how do they cause problems? And you mentioned accidental infections from non-human nematodes. What happens then?
Nematodes are truly astonishing. Their variety, their sheer numbers. They thrive in almost every ecological niche imaginable on this planet. Which, as you say, immediately raises the question, why do only a relative few parasitize humans? And what are the consequences when non-human ones take a wrong turn into us? So, descriptively, nematodes are non-segmented roundworms. Their bodies are smooth, cylindrical, not divided into segments like a tapeworm. And while there are thousands upon thousands of species out there in nature,
Like C. elegans, the labworm.
exactly like Caenorhabditis elegans, that's a perfect example of a free-living nematode that's taught us so much biology. Most nematodes are free-living, adapted to soil, saltwater, freshwater, you name it. Some are also parasites of plants, which is a big deal for agriculture. Now, the parasitic ones, the ones that infect animals, have generally co-evolved very specifically with their hosts. They usually can't complete their full life cycle in a different host species.
But humans sometimes get exposed anyway.
Uncommonly, yes. Humans can accidentally encounter infectious stages of non-human nematode parasites. And when this happens, these accidental zoonotic infections can cause significant trouble. Because the parasite larvae often can't develop properly in us, the unsuitable human host. They might migrate through tissues, triggering strong inflammatory and immune reactions before they eventually die off.
An inflammatory dead end for the parasite, but painful for the person.
Precisely. And we see some really striking examples of this. You might get pulmonary coin lesion spots on lung x-rays that can look like tumors caused by larvae of the dog heartworm, Durofilaria amidus, transmitted by mosquitoes. Or severe neurological conditions like eosinophilic meningoencephalitis, which can be caused by ingesting eggs of the raccoon roundworm, Balis ascaris porcionis, often from contaminated soil. Similarly, eosinophilic meningitis can come from ingesting larvae of the rat lungworm, Angiostrongylus cantonensis, maybe from contaminated snails or slugs or even produce.
Wow, so these accidental infections can be serious.
They definitely can be, and they require sharp clinical awareness. Now, shifting back to the nematodes that do establish true infections in humans, where do they live? They either reside within the intestinal tract, like the giant roundworm Ascaris lumbricoides, or they can localize outside the gut, in vascular or tissue sites. Think of the filarial worms causing lymphatic filariasis living in the lymphatics.
And how do they reproduce?
Most nematodes are bisexual, separate male and female worms. That's the standard setup. The big exception, which we flagged earlier, is Strongyloides stercoralis. The adult females of strongyloids living in the human gut are hermaphroditic. They have both male and female reproductive organs and can produce offspring on their own without needing a male worm in the host.
That's unusual.
Very. And depending on the nematode species, the fertilized females will release either live larvae directly or eggs that already contain larvae.
What about their development? Is it complex?
Yes, they have a structured development. 5 distinct stages. There's the adult stage and then 4 sequential larval stages, L1 to L4. Each transition often requires specific environmental cues or locations. Structurally, they're quite sophisticated for worms. They have this durable outer layer, the cuticle, which protects them. They possess a nervous system, a muscular system under the cuticle for that characteristic movement, and a developed gut, an oral cavity, an intestine, and an anal pore for waste.
And they vary a lot in size.
Dramatically. From tiny, barely visible ones to, yes, over a meter long. The guinea worm, Dracunculus medinensis, is the classic example of an incredibly long nematode. It just shows their amazing variability.
How do humans actually get infected with these various nematodes?
The routes of acquisition are highly varied, really depending on the specific parasite's life cycle. A very common way is ingesting eggs passed in feces, think Ascaris or pinworm, anaerobius, eggs contaminating food, water, or fingers. Another major route is infective larvae penetrating intact skin. This happens when skin is exposed to fecally contaminated soil. That's how you get strongyloids and hookworms. Big issue for farmers, kids playing barefoot.
The skin penetration, okay. What else?
Then you have larvae that enter through the skin via the bite of an infected insect vector. That's the mechanism for the filarial worms, mosquitoes, or flies transmit them. And finally, some important infections come from eating specific contaminated foods. Trichomoniasis is the classic example. You get it from eating raw or undercooked pork or meat from wild carnivores like bears that contain the encysted larvae.
So many different routes. And you mentioned internal reinfection earlier.
Yes, and it's worth reemphasizing this crucial point because it dictates so much about chronic disease. Only strongyloids, stercoralis, and Capillaria philippinensis can internally reinfect humans' complete cycles within the host, leading to potentially massive worm burdens without new external exposure. For all other nematodes, any increase in the worm burden must come from continued exogenous reinfections, new exposures from the outside world. This is fundamental for disease control. You have to break that external cycle for most of them.
That distinction about internal reinfection is incredibly important for managing patients and designing control programs. Okay, let's move to the second major group, the Cestodes. Tapeworms, these are really striking with their long segmented bodies. You mentioned they have a totally different survival strategy. How on earth do they function without a digestive tract? And what are their unique ways of causing disease? It seems they can cause trouble both in the gut and in tissues.
Exactly. What's truly remarkable about cestodes, the tapeworms, is just how extremely adapted they are to a purely parasitic life, especially how they solve the nutrient problem without a gut. And yes, that unique biology gives them this dual threat potential, sometimes causing relatively mild intestinal issues, other times causing severe, even life-threatening tissue disease. It all depends on which life stage you ingest.
Okay, so define the first.
Cestodes are elongated, segmented, hermaphroditic flatworms. As adults, they typically live in the intestinal lumen of their definitive host, often us humans. But, critically, their larval forms can live in extraintestinal tissues, usually in an intermediate host, leading to totally different problems.
And their structure, that segmented body.
Right. A tapeworm essentially has two main parts. There's the head, called the scolex. This is specialized for attachment to the gut wall, using suckers, hooks, or grooves, depending on the species. It anchors the worm. The scolex is also where new segments, called proglottids, are continuously formed. These proglottids make up the rest of the body, the strobila, and allow the worm to grow incredibly long.
And the nutrition part. No gut.
This is one of the most fascinating adaptations. Tapeworms have no functional gut tract at all. No mouth for ingestion, no intestine, no anus. Instead, each segment, each proglottid, absorbs nutrients directly from the host's digested food right through its specialized outer surface, the tegument.
Wow, they just soak it up?
They just soak it up. The tegument is highly folded to maximize surface area, making them incredibly efficient at passively and actively absorbing pre-digested nutrients. It's a brilliant parasitic strategy.
How do they reproduce them?
Mature proglottids are hermaphroditic. Each one has both male and female sex organs. Fertilization usually happens between adjacent proglottids in the same worm. These fertilized segments, packed with eggs, then detach from the end of the worm and are passed out in the host's feces.
Okay. Now, you mentioned two different ways humans get infected, leading to different diseases. This sounds really important for professionals to understand.
Critically important. These two pathways are distinct and lead to vastly different clinical outcomes. It's essential to grasp this. Pathway 1 involves ingesting larval forms in animal tissues. This is the common way to get an adult tapeworm in your gut. What happens is, a tapeworm egg gets ingested by an intermediate host, often a grazing animal like a pig or cow. The egg hatches in the animal's gut, releasing an oncosphere. This larva penetrates the gut wall and develops in the animal's muscles or tissues into a cysticercus larva.
A little cyst in the meat.
Exactly. Humans then get infected by eating raw or undercooked meat containing these cysticerci. Think Taenia solium from pork or Taenia saginata from beef. When we eat the infected meat, the cysticercus develops into an adult tapeworm in our small intestine. This causes intestinal toniasis, usually a relatively mild condition, maybe some abdominal discomfort, or just noticing segments in the stool.
Okay, that's pathway 1. Eating the larvae, getting the adult worm. What's pathway 2?
Pathway 2 is potentially much more dangerous. This happens if humans ingest the eggs directly.
Eating the eggs, not the larvae.
Correct. Eating the eggs, which are passed in the feces of someone who has the adult tapeworm, or in some cases from animal feces for certain species, this can happen through contaminated food, water, or poor hygiene hand-to-mouth transmission. In this scenario, the human acts as the intermediate host, not the definitive host. The ingested eggs hatch in our gut, the oncospheres penetrate our intestinal wall, and then they travel through the bloodstream and develop into larval cysticerci in our tissues.
Cysts in human tissues. That sounds bad.
It can be very serious. These larvae cause space-occupying extraintestinal cystic lesions in various organs and tissues.
Like where?
Classic examples include cysticercosis, caused by ingesting Taenia solium eggs. The cysts can form in the brain, neurocysticercosis, a major cause of seizures worldwide, or in muscles, eyes. causing neurological problems, blindness, pain. Another example is hydatid disease from ingesting eggs of Echinococcus species, often from dogs. These form large, slowly growing cysts, typically in the liver or lungs, which can eventually cause organ damage or rupture.
So the exact same parasite species can cause either a gut infection or a severe tissue infection, depending only on whether you ate the larva or the egg.
Precisely. This dual potential is not just biology trivia, it's a critical diagnostic and public health challenge. It means a doctor needs to ask different questions based on suspected exposure diet versus hygiene. The implications for the patient are vastly different. Understanding this distinction is absolutely key for any professional dealing with these parasites.
That's a profound difference, really highlighting how the stage ingested dictates the disease. Okay, one more major group to cover in detail. The Trematodes, the flukes. These leaf-shaped worms seem incredibly varied too, living in blood, intestines, liver, lungs. And you mentioned complex waterborne pathways. What makes their life cycles so intricate, often involving snails? How do they pull off these aquatic journeys?
Yeah, the Trematodes, or flukes, really are masters of complex life cycles, frequently centered around aquatic environments. And it raises that key question for professionals. How do they manage these incredibly elaborate cycles, often needing specific snails as intermediate hosts to ultimately get back into humans via routes like skin penetration or eating contaminated food? Trematodes are fascinating. Medically, we often group them based on where the adults primarily live in us. You have the blood flukes, the cystosomes, you have intestinal flukes like Fasciolopsis, and tissue flukes, which include liver flukes like Clonorchis or lung flukes like Periodontimus. This range shows their amazing adaptability.
And what they look like, you said leaf-shaped.
Often, yes. The adult flukes typically have a flattened, leaf-shaped body, and they have important anatomical features for their parasitic life: oral and/or ventral suckers. These muscular cups help them hang on tight in situ, staying put against the flow of blood, bile, or gut contents.
Do they have a gut like nematodes?
They do have an oral cavity and a gut, but it's a blind intestinal tract. It branches out, but there's no distal anal pore, so waste has to be regurgitated back out the mouth opening. And for nutrients, they use a dual strategy. They absorb some directly through their outer surface, the integument, similar to tapeworms, but they also ingest host tissues or fluids into that blind gut. So, unlike gutless cestodes, they have a partial digestive system complementing absorption.
How about reproduction?
Most trematodes are hermaphroditic. One worm has both male and female organs. They can self-fertilize or cross-fertilize if another fluke is nearby. The major medically crucial exception here are the blood flukes, the schistosomes. They are unique among flukes in being bisexual, separate male and female worms. And often, the male holds the female in a special groove, living in permanent copulation within our blood vessels. A remarkable adaptation.
Wow. And how do their eggs get out of the human body?
The routes for egg expulsion are also quite varied, and knowing this is critical for understanding transmission and control. Many important species pass eggs in the feces. This includes liver flukes like Fasciola and Clonorchis, the intestinal fluke Fasciolopsis, and two key blood flukes, Cystosoma japonicum and S. mansoni. But another major blood fluke, Schistosoma hematobium, is different. It passes its eggs primarily in the urine, causing urinary tract problems. And then you have the lung flukes, Paragonimus. Their eggs can be coughed up in sputum or swallowed and passed in feces.
So different exits mean different control strategies needed.
Absolutely. Improving sanitation helps for fecal-oral routes, but for urinary schistosomiasis, safe water for bathing might be more critical. A one-size-fits-all approach just doesn't work.
Okay, tell us about that complex life cycle, the aquatic part.
It's often incredibly intricate, involving multiple stages and relying heavily on water and intermediate hosts. Typically, it starts when eggs pass from a human reach freshwater. There, they usually hatch, releasing tiny swimming larvae called Myricidia. These Myricidia actively seek out and infect specific species of snails. This host specificity for certain snails is a really important bottleneck in their epidemiology. No right snail, no transmission.
What happens inside the snail?
Inside the snail, the parasite undergoes major multiplication and development through several asexual stages. One Myricidium can produce thousands of the next stage. Eventually, new larval forms called cercariae are released from the snail back into the water. These cercariae are the stage that infects humans.
And how do the cercariae infect us?
Again, it varies. For schistosomes, the cercariae directly penetrate the intact skin of people who are swimming, wading, or washing in contaminated water. Makes sense why schistosomiasis is common in communities relying on rivers or lakes.
Okay, skin penetration. What's the other way?
For many other flukes, the cercariae don't penetrate skin. Instead, they insist form a little protective cyst on aquatic vegetation or invade a second intermediate host. This encysted stage is called a metacercaria. Humans then get infected by ingesting these metacercariae. For example, you can get fasciola, liver fluke, by eating raw watercress or other aquatic plants with metacercaria on them. You get clonorchis, another liver fluke, by eating raw or undercooked freshwater fish containing metacercariae. And you get paragonomus, lung fluke, by eating raw or undercooked freshwater crabs or crayfish carrying the metacercariae.
This infection comes from contaminated water directly or contaminated food from the water.
Exactly. This heavy reliance on specific environmental conditions, water, the right snails, certain human behaviors like food preparation or water contact is fundamental to their transmission. It explains why these infections are often geographically patchy, limited to areas where all the pieces of the puzzle fit together. Understanding these complex pathways is absolutely essential for any public health professional trying to break the cycle.
That intricate dance between parasite, snail, water, and human behavior really highlights the ecological nature of these infections. Okay, we've covered a huge amount of ground, Trichomoniasis, the fundamental differences between protozoa and helminths, and then detailed dives into nematodes, cestodes, and trematodes. So bringing it all together, what's the bigger picture? What's the overall impact of these helminth infections, especially globally, and what are we doing about it?
Connecting it all back, yes. It's crucial to recognize that many of these helminthic infections fall under the umbrella of neglected tropical diseases, or NTDs.
Neglected. Why neglected?
It's a term that signifies, unfortunately, that these diseases primarily affect the world's poorest and most vulnerable populations, mainly in tropical and subtropical areas. And historically, they haven't received the same level of attention, research funding, or resources for control compared to diseases like HIV, TB, or malaria, despite causing immense suffering and disability. The health impacts are incredibly varied, but a key theme, as we discussed, is that for most helminths, significant disease burden often depends on that frequent need for repeated exposures to build up the worm burden over time.
Right, the cumulative effect.
Exactly. It's often not a single dramatic infection, but the chronic, insidious effect of harboring worms for years, often starting in childhood. This leads to a silent erosion of health, development, and economic potential.
What kind of specific problems do they cause?
In areas where exposure is common, especially from childhood, think soil-transmitted nematodes like Ascaris and hookworm, mosquito-borne filariasis, or waterborne schistosomiasis, the morbidities are profound and wide-ranging. We see major nutritional impacts: anemia from hookworm blood loss, malabsorption, competition for nutrients. We see impaired physical growth and cognitive development in children, affecting school performance and future opportunities. And various functional impairments, the disfigurement and disability from lymphatic filariasis, the bladder cancer risk from S. hematobium, the liver fibrosis from other schistosomes.
So they contribute to the cycle of poverty.
Absolutely. Chronic helminth infections can significantly undermine health, education, and productivity, trapping individuals and communities in a cycle of poverty and poor health for generations. It's a massive, often underappreciated burden.
So what's being done on a global scale?
Recognizing this huge impact, there are major ongoing global programs focused on controlling these diseases. A key strategy is mass drug administration, or MDA. This involves periodically giving safe, effective anthelmintic drugs, worm medicines, to entire at-risk populations, school children, sometimes whole communities, regardless of whether individuals are known to be infected.
Treating everyone in an area.
Essentially, yes, in high-risk areas. The goal of MDA isn't necessarily to cure every single infection, but to dramatically reduce the overall worm burden in the community. This lessens the morbidity associated with heavy infections and, crucially, reduces the amount of eggs being put back into the environment, thereby interrupting transmission over time.
Has it been successful?
These programs have had considerable success in many places, significantly reducing the prevalence and intensity of infections like lymphatic filariasis, onchocerciasis, schistosomiasis, and soil-transmitted helminths. Morbidity has decreased dramatically in many areas. However, there are ongoing challenges. Sustaining funding, logistical hurdles in reaching remote populations, ensuring community acceptance and participation, monitoring for potential drug resistance. It requires long-term commitment and integrated approaches, often combining MDA with improvements in water, sanitation, hygiene, W-wash, and vector control. The global professional needs to understand both the power and the complexities of these large-scale interventions.
It's abundantly clear then. Understanding these parasites down to the molecular and ecological level isn't just academic. It is absolutely fundamental for designing, implementing, and sustaining the public health strategies needed to lift this enormous burden from millions worldwide. From Trichomonas to tapeworms, each requires a tailored, knowledgeable approach. We have really journeyed through an entire hidden world today. We started with the specifics of Trichomonas vaginalis, its prevalence, those diagnostic challenges, the absolute necessity of partner treatment for control. Then we shifted gears, defining those fundamental, crucial differences between the single-cell protozoa and the multicellular helminth structure, replication, those distinct immune responses like eosinophilia. That foundation is key. And from there, we dug deep into Chapter 230 territory, exploring the major helminth groups, the incredibly diverse nematodes, the roundworms, highlighting zoonotic risks, and that critical concept of internal reinfection being rare. We examined the intricate cestodes, the tapeworms, with their gutless existence, and that shocking difference between intestinal disease from larvae and severe tissue disease from eggs. And finally, the very trematodes, the flukes, unpacking their complex, often aquatic life cycles involving snails and their diverse routes into humans via water or food. Getting this comprehensive picture from the worm's structure to its global impact is what truly marks an elite professional understanding.
It really is. And this incredibly deep dive into their biology and epidemiology, it naturally leads to a forward-looking, maybe slightly unsettling question for us all to think about. As our world changes, as global travel becomes more common, potentially moving parasites or exposing people in new ways, and as environmental shifts, changes in water patterns, temperature, land use, maybe climate change itself, alter the habitats and ranges of those crucial intermediate hosts, like snails and insects, how might the epidemiology, the geographic maps of these complex helminthic diseases shift in the coming decades? What new challenges will emerge, and how will our deep professional understanding need to adapt to create effective public health strategies for a changing planet?
That's a powerful thought, a really provocative question that underlines why continuing to study and understand these organisms is so vital. The landscape is always shifting. Thank you for joining us on this deep dive today, and thank you, our listener, for your commitment to understanding these complexities. Keep exploring, keep asking questions. We look forward to our next deep dive together.